Good morning, everybody. I'm Susan Whitehead. I've been a longtime board member of the Autism Consortium, and it's really a privilege today for me to introduce Dr. Rudolf Jaenisch, our first speaker. Rudolf is a founding member of Whitehead Institute and a longtime professor of biology at MIT. He's here for a number of reasons because he's been a pioneer in a number of fields. I think what's most relevant for today's meeting is that he was one of the founders and the first person to develop a transgenic mouse used in mouse models of human disease, which was really a tremendous breakthrough. The second thing that's so important that Rudolf has done is he's one of the founding fathers of stem cell science in the world. He works in both embryonic stem cells and induced pluripotent stem cells. And I see a lot of parents in the audience. I'm just going to do a quick translation. Rudolf knows how to make adult stem cells behave like embryonic stem cells, which is essential and very important work. He also is the current president of the International Society of Stem Cell Research, and you may have heard his name before in the public because in 2011 he received the National Medal of Science. I'm not going to say anything else except one personal comment, which is that I've known Rudolf for over 30 years, and I have a very clear memory of when I first met him. He walked through the door, he was on fire. Anybody who knows Rudolf will know what I'm talking about. He has such a zeal and passion for his research. It's 30 years later, and that remains completely undiminished in Rudolf, and his contributions remain completely undiminished. It's just been a remarkable journey to be witness to. Dr. Rudolf Jaenisch.
Well, thank you very much, Susan, for this very generous introduction. And thank you for inviting me to this meeting. So what I would like to do is to talk on science, not really on the clinic, but on more the basic science, which I think allows us to make progress in the clinic. So the promise of embryonic stem cells is really tissue engineering. You can make any cell type of the body from an embryonic stem cell. And this, of course, is really of major potential significance. So the goal of stem cell research is to use the potential of these cells to generate genetically defined models of complex diseases. That's what I want to focus on, and then eventually do it with cell therapy. So the approach involves the conversion of somatic cells to a pluripotent state, and this is being done by three ways, either by nuclear transfer, as done by half a century ago by John Gurdon, and now by defined factors by Shinya Yamanaka first in 2006, which really led to the explosion of the stem cell field. So the concept of using these pluripotent cells for disease research is very simple. It is really using, if you have a patient and you want to study a certain cell type which is deficient, you use the Yamanaka approach or other approaches to generate really the pluripotent ES cells or iPS cells, and those become then differentiated to the cell type you want to study. You can do this also directly, but the cell type you would want to study, that's what you use to model the disease. So there are a number of key issues in the field which I want to highlight. These are important if you want to make that system really relevant and use its full potential for studying complex human diseases. One is the nature of human pluripotency, the quality of embryonic stem cells or iPS cells, how good are they, what are the criteria, how do they compare with mouse cells, maybe you can do all these assays, and are iPS cells different from ES cells? Well, iPS cells are the ones which are derived from a skin cell, let's say. I think genetic manipulation is extremely important. And finally, mechanistic understanding of complex diseases is one of the goals. So I'm not going to talk about the clinic. I'm going to talk about some basics. And I want to concentrate on two topics. One is the nature of human pluripotency. Now, this is maybe a little bit esoteric for many of you, but it's a very important issue if you want to use it. And I will come to this, why I think that's so important. And it's called the naive versus the primed human cells. I will define this later. And in my second part, I want to really give you some concrete examples of how we can use this technology to study autism. So let me go to the points here. You can make pluripotent stem cells from the inner cell mass of the blastocyst or from a later stage of embryogenesis. They're called ESC cells. Both of those are pluripotent, but actually they're very different. So this is in the mouse. Now, so in the mouse, if I go back, you make ES cells and IPS cells, or you make these later ESC cells from the implanted embryo. The origin of ES cells is the inner cell mass of the embryo or the somatic cell. ESC cells is the epiblast. In human, you make ES cells and IPS cells. They come from the inner cell mass or from somatic cells. But they're very different. Those cells are small and dense. Those are flat. The mouse ESC cells as well as human ES cells. They have different growth requirements. I don't want to go into details here. And the X inactivation, something which happens in a female embryo, it's prior to X inactivation in mouse ES cells. The human ES cells are post-inactivation. They're further along. The state of pluripotency can be best measured in the mouse by generating chimeras. The ESC cells cannot make chimeras, and human cells, can they make chimeras? I will come to that. That's a big issue. So these two states have been called the primed state, more differentiated already, from the naive state, the more grown state, the inner cell mass, embryo-like state. So the point I'm trying to make is, so here are we. So the ESC cells are made, called the primed pluripotency, and by defined factors, we can convert those in the mouse to naive pluripotency. However, what I want to concentrate is, can we do this with human cells? Does the state of pluripotency exist in the human? I will come later tell you why I think that is important. It's not only esoteric. So let me just define it. They're different states. Most naive cells are unlike most ESC cells or human embryonic stem cells. They're the primed ones. But you can convert them by different cytokines from one to the other in the mouse. Can you do this with human? That is the question I want to discuss now. So there have been a flood of papers. It's a very controversial issue, a flood of papers which say, yes, we can make naive human ES cells. And all these papers used, and they're in very high-profile journals. One came out just last week in Nature. They all used candidate approaches, some thinking about what is important. Our goal was not to have any preconceived notion, but do it in an unbiased way. And this was done by a postdoc, Thor Teunissen, and a student, Ben Powell. So we used a reporter system, and it's based on OCT4. OCT4 is a pluripotency factor, which is expressed in ES cells. It's a critical factor. And its expression is driven by what's called a proximal enhancer in the primed state and by a distal enhancer in the naive state. So using now gene editing, and I want to go into this, we generated an allele which is labeled by GFP and has deleted the proximal enhancer. So these cells are green if they carry this element, but they're becoming dim when you delete it. So that's the basis of the screen. So the screen, you can use two transgenes, KLF2 and NANOG. These are pluripotency factors. I will not go further into those. If you put those into these cells, we can activate this reporter, as you can see here with GFP. And you can see this here. These cells become GFP positive. Before, they were not. When you withdraw, this is an inducible system, and you withdraw doxycycline, turning off these transgenes, the reporter is silenced, and the cells really do not express GFP anymore. So we know now this is a system where we can screen now what maintains naive pluripotency, and we did that by using really these cells, convert them to GFP positive cells, then withdraw, inactivate the transgenes, and now ask the question, what maintains the expression of the reporter, and will not go into the screen. But what came out of this was repeatedly hitting a BRAF, which is an oncogene, it's a kinase, and if you inhibit that, then you maintain the reporter activity. So BRAF inhibition maintains this reporter activity in the absence of the transgenes. We can also ask the question, can we, without any transgenes, convert cells to the naive state? And we can. And I will not go into details here. We can do this. We can define these conditions. And the final question was, can we make new ES cells from human blastocysts, from human embryos, under those conditions? And we can. So if I summarize this type of screen, it is these, we call it 5I conditions, 5 inhibitors that can maintain reporter expression and can generate new ES cells. So what does it mean to have the reporter on? Does it tell us anything? So how do we really assess pluripotency in humans? In most, you can make chimeras. You have all these assays. In human, you don't. So gene expression is one. You can look at gene expression profiles, report activity, the chromatin state, and a developmental potency, which means in vitro differentiation, teratoma formation, and chimera formation. So I will come to these issues. So just very briefly, this is an expression profile of mouse naive cells. These are our naive cells. They're very close to those. And this is an assay which I really don't want to go into this audience. It's called a principal component assay, which argues these are our starting cells in this space. These are the naive mouse cells. These are human embryos. And all of these papers which I used before really overlap with the primed state, whereas our cells, as well as those of a paper published by Austin Smith, are very close to the naive mouse ES cells. But I really don't want to dwell on this. One question was, do these alternative papers, do they activate the methods described in those? Do they activate our reporter? None of those does, whereas our conditions do. So these are quite some differences. And I will polycomb-targeted chromatin, I think I will skip that. But I want to come to the developmental assay. How do you measure in human cells the state of pluripotency? What is their potential? That's an important issue. So you have several ways to do that. In vitro differentiation, you take the cells, differentiate them to neurons. Can they do it? Yes, they can. Teratoma formation is more stringent. You ask these cells to make a differentiated tumor in the mouse. They can that. The more stringent assay is, of course, chimera formation. Now, this means, can these cells contribute to the development, let's say, of a mouse? And this was actually published in one of these papers, in Gaffney et al. Last year. So we asked the question, can our cells do that? Now, this is a complicated assay, and I will just summarize that. We injected cells which were labeled with the GFP or with a red marker into different stages of embryos. We used the cells published in this paper, and you can see quite a lot of embryos were injected. There was some cell death. And then we asked the question, was there any evidence for chimeras? And for this, we used GFP. Do these embryos carry the GFP marker? And when we do this, none of those did. Now, this is not a very sensitive assay. So we asked the question, can we use a more sensitive assay? We used a genetic assay or DNA analysis using human mitochondrial DNA. And there we can detect less than 1 in 10,000 cells. Now, this is our detection limit. So none of these embryos actually had a chimerism of more than 1 in 10,000 cells. Basically, they were all negative. So the conclusion from this is that we cannot really, under those conditions, generate chimeric animals as was reported in this paper. So clearly, this is not what we can reproduce, at least. Well, question of axon activation, as I said before, in the mouse, axon activation in the embryogenesis occurs later, after the blastocyst stage. Both axes in female embryo are active. Naive cells, both are active. In prime cells, they have undergone axon activation. But when you convert these cells in the mouse, the prime to the naive, they reactivate the inactive X. Very clear data. In human, this is very, very confusing. In human, the paper in 2011 from Edith Hurt has shown that both X chromosomes really exist, which is the marker for X inactivation. Conventional ES cells really are XA-XI. They have undergone X inactivation and some other exceptions. So the question we asked, when you convert these prime to naive cells, do they reactivate the X? And this is now an assay, which I just briefly say, you look throughout the X for expression in a male which has only one X that is set as 1, and you can see our starting cells had both X chromosomes active, but when you convert them to the naive state, they inactivate the X. This was very surprising. That was totally counter to expectation, what we see in mouse cells, where we, of course, convert the conversion of prime to naive cells really induces X inactivation instead of activation as in the mouse. So we are confused about that. That's very different, but the human is very different from the mouse. And of course, the question is really now, what is the key, what is the ground state of X inactivation of pluripotency? So let me summarize that part of my talk. So these 5I conditions, and these are the inhibitors, really activate this reporter. They convert the ES cells to a naive state as compared with the mouse. And we can make new ES cells, and they really have all signs of the naive state which corresponds in the mouse. So clearly it's a distinct state of pluripotency that shares defining features with naive mouse ES cells. And the question is there only one state of pluripotency in the human or not? And so there may be more. But now we can ask the question, why should we even care about naive cells? We have the primed cells that can do everything. Why bother? Is it all well, esoteric interest, academic interest? So let me give you my view on that. The approaches to model diseases in culture, really they have limitations. And these limitations sometimes, can we really study complex diseases such as autism in a dish? And these are questions which really can keep you sleepless sometimes. So what we study, what we use is really a culture system. We use a 2D culture system to study these differentiated cells, to study the mechanism of disease, which means we start with human ES or iPS cells and differentiate those to the differentiated cell type we want to do. Now there's a lot of limitations in this two-dimensional system. Of course, most protocols yield immature cells. These two-dimensional cultures do not recapitulate life. Life is, of course, in a three-dimensional space. Cells live in this 3D space. And you cannot study really easily cell-cell interactions or cell migration, very important issues of the of studying a disease. So what are the potential solution for this? It would be an in vivo approach, which I think would involve human-mouse chimeras. Now, many of you might think this is a complicated and maybe controversial assay, but I want to say that you can make the chimeras by injecting a cell into a blastocyst, which I showed you was totally unsuccessful so far, or taking committed cells, neural precursor, put them in utero into the embryo, and really have the goal to generate a mouse where part of the neurons in the brain of the mouse are derived from a patient which has, let's say, Parkinson or Alzheimer, to study this in vivo. So that's certainly a goal. I think it's a very high bar, but I think that's what, from my point of view, justifies to work with to try to define the naive state which could possibly do this. But this is a very high bar, and we're trying to solve some of these issues. So let me come more to the concrete issue of this symposium, namely to autism. So autism, most autism cases are sporadic, and we really don't know the genetic underpinning. But there's some which are monogenetic, and they have high rates of autism, and there's some examples here like Fragile X, and quite a number of others. We are interested in Rett syndrome, in autism and in SHANK3. And I will really talk only about Rett syndrome because that's a more defined story. And what we want to do is to make an iPS cell-based modeling of this severe autism. Now, Rett syndrome, many of you are probably familiar with it. Let me just recount it. It's a terrible disease. It only affects girls. Boys die because it's an X-linked disease, an X-linked gene which is mutated, MECP2. So they go into this regression phase at 1 year of age. When they lose all speech they might have, they become very highly mentally retarded, no speech. They have these stereotypic hand movements, and they have a smaller head. Now, this was a very interesting disease. We made the mouse model of this now more than 15 years ago, before MECP2 was really identified as a cause of Rett syndrome. And the mouse really very well mimics the human disease. And I give you one example of this. If you lift a normal mouse at the tail, they spread their paws. If you lift a Rett mouse, they clutch their paws. So it was very surprising. Many, many other issues were there found. Now, this gene, MECP2, which is mutated, has been shown to be binding to methylated CBG and recruits co-repressors, basically silences genes. The problem with this concept was, when we looked at the mutant mice, there was really not a global activation of genes as you would expect from this concept. It was very minor. It was really somewhat disconcerting. Rett syndrome is called by clinicians often a developmental disorder. This might not be right because of these studies. If you have a Rett mouse which is about to die, very seriously sick, and you re-express the wild-type gene in their neurons, they get rescued. So this gene is important for the maintenance of neuronal function, not for the development. And we have done actually another experiment where we express BDNF, a cytokine, in the brain of these animals, and that could also rescue. Now, in a clinic, you cannot express the gene in the brain of a patient. You cannot introduce BDNF. So what is the clinical relevance for this? Well, a couple of years ago, we made a study together with Mirkan Kasur at the Picaro Institute, where we used IGF-1, which is cytokine, injecting this into the mice, in rat mice, and we showed that this could halt the disease. Now, this was more interesting, but IGF-1 is an FDA-approved drug. And based on this study, a clinical trial was started in children's, and the results were just published. And I think they were highly encouraging. There was no problem. And they could measure, they could see an improvement of quantifiable problems in these children, such as autonomic dysfunction. Now, I think this is very exciting. And for me, that's really very satisfying, because a mouse model which we made 15 years ago and led directly, the study of this led to know something which is a promising clinical trial. So, what I want to do, model this Rett now not in mouse but in human cells, and this was done by these two postdocs in my laboratory, and we made a Rett mutation in human cells. We generated isogenic pairs of cells which either had the functional MECP2 or not, and we wanted to see, can we see a phenotype in the culture dish? And when we culture these iPS or these ES cells to neurons, you can see that the mutant cells are much smaller than the wild type. You can see this here. And when you look at their arborization of their neurites, they're much less complex than in the wild type. And you can see this here quantified here. So they have a very drastic cellular phenotype, which you can see. We can also look at a functional assay by plating these cells on a microelectrode array. Now, this allows you to record electric activity. And so you can plate mutants and wild-type in the same array, and then you differentiate this to neurons, and you can see a wild-type neuron is very active electrically. The mutant is more or less dead. So the Rett mutants have smaller cell size, reduced complexity, and reduced electric activity. Now, I could go into revisiting gene expression, and we did that, and I think because I'm running out of time, I will quit that, come rather to some other experiments. I will just go over this. What was important for us was a study from Rick Young, who argued that all the expression analyses we did previously were possibly flawed because the cell size was different. I will not go into the details here. And when we looked at RNA content in mutant and wild-type cells, you can see the mutant cells have much less RNA. Mutant neurons, but not neural precursors, have less total RNA. So when we now do an experiment for gene expression directly looking, comparing the number of cells put in the test, then we find actually that this gene doesn't act as a repressor, but acts as the opposite, as a global activator. It's totally unexpected. And I really didn't very well explain that, why we came to this conclusion, but it's a very firm conclusion. The genes which are expressed or which are affected by MBCP2 are particularly genes which are very highly expressed. They are reduced, whereas the low expressed genes are really not affected. So actively transcribed genes are preferentially downregulated in these mutants. And also genes which are important for ribosome, for protein synthesis, and protein synthesis is affected, and mitochondrial function is affected. So these cells, the mutant neurons, have major metabolic differences from the wild-type cells. Now, I told you that BDNF and IGF-1, when you treat the mice with these two cytokines, it can rescue the phenotype. That's in the mouse. Can we do this in these mutant neurons, where we have a clearly defined phenotype, which we can measure? And for this, we differentiate these neural precursors to neurons in the presence or absence of these cytokines. And what you can see is here that this is the control, this is the mutant. And when you culture those in the presence of BDNF or IGF-1, you rescue the cell size defect in the mutant. You can do this also by inhibiting this pathway, this mTOR pathway. And when you do that, you rescue the soma size, and even you rescue the dendritic, the less dendritic arborization of these cells. Protein synthesis is also affected. So let me just come to the end. So P10 inhibition, BDNF, and IGF-1 treatment rescues the defect in cell size, in neuroid complexity, and in protein synthesis. So what we think is happening, if this is a wild-type neuron with a certain complexity, certain amount of RNA and protein, then in a RETT neuron, it's smaller, they have less complex, they have less RNA and less protein. Now you can certainly convert this to there, or you can rescue this by BDNF and IGF-1. Now this is a very robust cellular phenotype in culture, as I showed you. So our goal is now to screen, or using the system to screen for factors that prevent, delay, or even reverse the phenotype of RETT in the culture dish. This would be, of course, of great interest for us for a clinical application, which might be more efficient than IGF-1. So let me come to the end. Disease modeling, the issues. What are the issues? I think iPS cell generation is important. We need genetically unmodified cells. I think this is resolved by using RNA or non-integrating viruses. What is the difference between IES and iPS? I don't think that's a key issue. Genetic manipulation. There's enormous progress now. We can do anything now in human cells using the CRISPR, TALEN, and zinc finger technology. I think differentiation is the main problem. It's the most important problem, because often you don't get mature cells, immature cells in blood lineage and in the endoderm. And if you want to use it for transplantation, there are major issues. What are the cells you want to transplant? Stem cells or mature cells, and where do you put them? And clearly, easy would be hematopoietic cells, that cells know where to go to correct a defect in, let's say, in blood, like sickle cell anemia, except we cannot make mature blood cells, and they wouldn't engraft. Pancreatic diabetes 1 would be treatable. I think, again, the problem is mature cells, although there's some progress now. Liver cells, the same thing. We cannot make mature liver cells. Probably the very first application of transplantation will be the retina, macular degeneration, where people have shown we can make mature retinal pigment cells, which can be in their clinical trials are starting. So I think the concept of having a patient generating patient-specific iPS cells to use this for modeling disease, I think we're very far advanced on this. I think more issues are have to be resolved to use it for transplantation. So let me come to the end here. I mentioned all the people who did most of the work, Teunissen, Ben Powell, and Yun Lin and Jun Muffat. We collaborate with Rick Young at the Whitehead and with Megan Kasur on the Rett syndrome, and with Natalie Gray at Harvard on these naive cells. Thank you very much.